we are going to study E. coli and non-resistant tuberculosis bacteria, and we are looking at multiple COVID-19 virus variants. And the first 2 days, it is the wet lab part. It means that we are extracting the genetic material and we are just estimating the purity of it, purify it, and prepare it for the next generation sequencing. It takes 2 days, and then at the end of these 2 days, we have the raw data from the next generation sequencing machine, and then we go from there to bioinformatic parts. Bioinformatic part, it is a little bit more elaborated in the sense of it has more theory on it, and during these 3 days the people, they learn about the bioinformatic by itself, and they learn also how to apply bioinformatic for these smaller genomes and how to come to a result, which in this case it would be that we could recognize that our sample, they were E. coli, or they were the non-resistant, say that TB, or what variant of COVID virus it was there, and if there was some mutation, or it was the original sequence of Omicron.
So this workshop has really been an intensive and educative one. It's a 5-day workshop where we, the participants, were taken through bioinformatics and also the wet lab preparation. So we participants had an opportunity to extract nucleic acids. We did library preparation, after which we sequenced our data.
Now, science is not all about lab and field work. There's more to science than that. So, aside getting to the lab and running your samples and getting results, you have to read meaning into the results obtained. So, this training had to do with the sequencing part and also the informatics or bioinformatics part.
I think, me personally, this is important to me because this is a field that I'm really interested in, and I find myself reading a lot of articles regarding bioinformatics or bacterial genomics, and I don't really have an understanding of it, but this is going to help me to really appreciate the papers I read and to be able to use it for my future applications. And then to my group, I think for most of the times, these are something we want to bring on board, and because we lack the expertise, we are unable to integrate it in most of the projects that we do.
These are very complicated technologies, and we are in the position now here to cope with these kind of technologies. These technologies, it can also empower us that to go further for further diagnostic. It doesn't need to remain too limited to the pathogen here. It could be for detecting also different kinds of cancer, different kinds of diseases that one can use genomic technology for detecting them.
I believe that when this workshop is open to more people, we can increase our knowledge, and also people can have a feel of genomics and bioinformatics.